Only two bits left, really, the immune system as such and vaccines and everything surrounding the vaccines. So immune response, here we go. Your specification with the EDUQAS is quite specific. Please do not follow anything that AQA and OCR have put on because it follows a slightly different pattern, so careful with that. You will need to know the difference between the innate and the adaptive immune system. So picture this a bit like a flow chart, immune system splitting up into innate and adaptive. And then the adaptive immune system, again, can be split up further into how it's responding in a humoral response and the cell-mediated response. So fluid-based with regards to proteins and cell work themselves. We can then also, and we need to move on to that, if you're exposed to some pathogen a first time round and a second time round, how does your primary and your secondary response differ? There are quite some fun graphs to go with that, and I've got no idea what maths work they might want you to produce on that one, but we can see if we can find some past papers on that sort of maths work. I've got one graph that I want to look at with you as if you were the examiner. So let's start with splitting it up into your natural disease resistance against any potential pathogens, against anything you were to potentially have. You've got your natural barriers, your innate immune system. It attacks anything. It doesn't matter whether it's chickenpox or whether it's AIDS or whether it's a rhinovirus or hay fever. Anything is being attacked, so literally pollen, anything is initially being attacked in a similar way. For the hay fever sufferers, of course, that is. And then you've got the adaptive immune response, which is slower because cells and proteins need to be built first, need to be dividing first, so that takes some time. However, that one is specific to that particular pathogen. So let's first do the innate immune system. What have you got? The first problem that any pathogen is going to have is how do you get in? Skin can't get through, it's waterproof, it's impenetrable. Keratin is a bit of a tougher protein, collagen is a bit more wobbly. You've got elastin in your skin as well. Various proteins are just make an impermeable layer unless you poke a hole through it and you're a mosquito, you've got a pin or a needle or something. The next thing you've got on the surface of the skin, you have got the skin is actually, you know the epidermis, epithelial cells. You've got many layers of epithelial cells. The top layers always come off, new layers are built underneath, pushing upwards. So whatever fungi might want to try and grow on the surface, they just get shed and washed off in the shower. Also, you have got your normal bacteria flora there. Your healthy bacteria will always outcompete any pathogen given the right conditions. Obviously, if something is wrong in that balance, then pathogens might take over and you might get a skin infection. Going a little bit deeper, still partly physical. In the lungs, you've got the physical trapping of sticky mucus and the ciliated epithelial cells to sweep that mucus back up, and usually you swallow it or you it kind of comes out. To go within the lungs, you've actually a generic enzyme cocktail that will just attack whatever is there, mainly proteases, to start digesting away on whatever cell walls you've got on bacteria. Then you've got blood itself. If capillaries are broken, then clots form right away and stop bacteria or anything else from entering. So literally platelets, the thrombocytes, will let their fibrinogen turn into fibrin, and you've got all those strings and it's trapping all the cells. Just taught in the circulation transport unit. Also, another thing that blood can do, it can cause an increased blood flow, and it will cause that whenever something seems wrong, an increased blood flow to the site will happen. General inflammation, general excess blood flowing in between the tissues of the interstitial fluid will be pushed around faster with the idea to have white blood cells to go between cells and see what there is. Can be from a twisted ankle, can be from an infection. Okay, this is a generic, inflammation is a generic response. You may have heard the word histamines as in an immune system communicator in how to cause inflammation to come about. And obviously antihistamines would do the opposite. Now, if you have inflammation as well, you are usually feeling that the area is not just swollen, but it's hot. That is making it a bit awkward for bacteria to reproduce if it's too warm. You then also have because it's non-specific. The innate immune system doesn't only have physical barriers, and you've got actually a whole lot of list of chemical barriers. You've got all your acidity in the stomach, you have got your various enzymes in random places from your tears and saliva. You've got various enzymes there as well, that's chemical barriers. But you've also got a few cells, all your bigger white blood cells, the macrophages and neutrophils. They do their phagocytosis on anything that has not got the self markers on them. However, it's a bit tricky when we're talking about things like allergies, because that is a maturing of the immune system later on. But for now, let's think of it as they're attacking anything that hasn't got the self antigen on the surface. Just a little side note, if you're thinking of phagocytosis and flowing around something, macrophages and amoeba have got a similar way of moving with the using of their cytoskeleton. But that kind of begs the question if there is an evolutionary link, even though they're moving similarly, apart from being eukaryotes, there seems to be not much there. So now moving from the innate immune system to the adaptive. So this is now where we're considering, right, we need to have a specific response for a particular named pathogen, or not named, but literally one specific one. There is a whole range of body parts involved, of organ parts involved in this system. Generally, you will need to know that the bone marrow is the site of production of your red and white blood cells. So the production, however, they are going to be maturing somewhere else as well. So the thymus is going to be involved in the maturing, the spleen is involved in the maturing. There are various body parts of your immune system where you've got a higher density, lymph nodes and so on. There's a huge system to go with the lymphatic system as well. So not only the lymph vessels that you've learned about, but the lymph nodes and aggregations of nodes into bigger systems, let's say, in your digestive tract or your armpit. And the problem is sometimes that self and not self is being misinterpreted, and something goes wrong somewhere along the way. There's a lot of research taking place into when and where autoimmune disorders develop. Now, I will leave it as that. I just wanted you to have a bit of a list of where the immune system is starting to attack itself. Think of this as if you were to have a hay fever-like response instead of to pollen. You've got a similar response to your own tissue, and it tries to break your own tissue down. Let's leave that as that. Right, the adaptive, that can be split up into two sections. One of them, think of the humoral response as anything that is flowing in a fluid. So the antibodies, which are basically bigger blood proteins, very specific proteins flowing in the bloodstream in the actual plasma. And what they do, so we'll get to that. The cell-mediated response, cell says it itself, everything that's to do with cells. And obviously those cells will then have an impact on the humoral response, because you can't just have proteins made out of thin air. You will need cells to make those antibodies, to make those proteins. So it is flowing hand in hand a little bit. We'll look at the humoral response first. So again, here we need the cells to make those antibodies in the first place. B lymphocytes, they're made like all white blood cells in bone marrow, but then need to mature in the spleen and lymph nodes, where they're being bombarded with various information about the body, what cells are self and what cells are not self, what cells are acceptable, what cells aren't. Most of that is being done before you're even born, some of it shortly after birth. So your immune system does still respond as it grows, as you go older and age. Now, when and where, whenever a foreign protein is being detected, they divide and make two types of cells. Now, for B cells will go after the actual pathogen itself. They will recognize the pathogen. They will make antibodies for the actual pathogen. You'll hear of T cells later on. They do a similar system, but going after the infected body own cells. So not the actual pathogen, but the actual self cells that have become infected. So if you look at the diagram, if a particular protein has been recognized by one of a few million of B lymphocytes, you've got colonies of lymphocytes, you've got a range of different ones. If you happen to meet the correct one, we've got literally B lymphocytes waiting for infections that you'll never meet in your life. You've got plenty of spare. So if you were to meet a surface protein that was looking pathogenic, that is not me, that particular B lymphocyte is being given the information to, right, you now need to divide, 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 divide. It produces 2 different types of cells as it divides and forms various clones. Clones because it's got that particular antigen that it responds to. The plasma cells, they will then make the antibodies, and memory cells. We will get to those when we're looking at vaccinations and how your immune system builds up over time if you're exposed to an infection a first time and then maybe a second time and a third time. So the plasma cells we're going to focus on now with regards to what is happening there. Plasma cells produce antibodies. There are various diagrams of antibodies, and yeah, they're called immunoglobulins as well. Sometimes they don't really look like that. Why? They look more like a inside out pyramid, something weird arrangements. But for your textbook, the Y shape is what you need to have. They're glycoproteins, so mostly protein with a bit of sugar attached to it. The structure of them is quite sweet. So you've got your polypeptides, you've got heavy parts and light parts to go with them, and stabilization with a couple of disulfide bridges to stick them together. You've always got constant parts to that. The bottom part of it seems to be a constant part that is always there for the antibody. However, you have got a specific, a variable region. Now that is created by genetic shuffling. So your genes just do something random and just flicker about, and let's see what different amino acids can we put together here. Let's do a bit of randomness and kind of throw up all the nucleotides in the air and see what happens. So you're producing literally a random shuffling here for that variable region. The variable regions being at the top of that Y. And that is where you have your binding site to whatever you could potentially be meeting, the antigen binding site. Think of that as, I don't know, your 2 million different shapes that it can attach you to. Very similar to the enzyme substrate fit that you've learned about with the lock and key model versus the induced fit model, so that you've got the shapes matching and flowing into one another. Now, antibody, the definition is it's an immunoglobulin responding in response to an antigen being presented. So an antigen is the protein that might be on your bacteria or virus, your foreign protein, that's your antigen. And the immunoglobulin is the protein with the heavy chains and light chains, with the variable regions and the constant regions, that will then be manufactured by your B lymphocytes in response to that antigen. Now, what do? They will stick themselves together and onto your pathogens. If you have an antibody with that particular shape that you'll find on your pathogens, so those purple things are supposed to be bacteria, you've got your antibodies here, they will stick them together. They will also tag the bacteria for a phagocyte, for a bigger white blood cell to come along this is what you need to gobble up. This is where you need to do your engulfing party. So it's agglutination, sticking together, and then making it easier. If you've got a bigger lump for your phagocytosis, a bigger number of minute cells lumped together, it's easier for the macrophages and neutrophils to flow around that, engulf that, and then digest it. Now, I think I've mentioned hemagglutination when talking about blood types, so that if you were to have, let's say you are blood type A and you've got blood type B injected into you or infused into you, that your blood would literally be lumping because your antibodies would recognize the foreign blood and stick the cells together. So hemagglutination in that context. Right. Why do we need those variable regions? This is now the fun part. You're actually born, even though during your pregnancy, while you were pregnant, during your pregnancy, you had loads more plasma cells and loads more B cells that had various gene regions, random genetic combinations. But your B cells need to be killed off. It's a little bit of if it's attacking your own cells, you need to delete those B cells because they're attacking self. And that is what's happening long before birth, but it does go into early childhood as well, that you've got a deletion of, okay, it's just in the air anyway. It's supposed to be that if you're early on in your childhood are being exposed to, I don't know, to peanuts and to hay fever pollen, that you are recognizing, or your immune system recognizes, okay, that is all fine. I do not need antibodies against that. Therefore, the B lymphocytes for that can be killed off. They don't need to be existing anymore. However, clearly it doesn't work perfectly, and autoimmune diseases exist, allergies exist. Yeah. The other response, the cell-mediated response. So when exposed to pathogens, so when exposed to a foreign antibody, sorry, when exposed to a foreign antigen, you've got the activation of all your bigger white blood cells, your phagocytic cells, your bigger white blood cells that go around engulfing things and eating them up, plus your B and your T lymphocytes. Your B lymphocytes, they were the ones that, if they're selected because they've met the right antigen, they're turning into plasma cells. Those plasma cells then proliferate and produce loads of that version of antibody, but also into memory sites. The attack goes straight away against that antigen. So you're going to find that on the actual bacteria, on the virus, and yeah, agglutinate those together, stick those together, deal with them directly. T lymphocytes, they are not maturing in the spleen. Again, T lymphocytes are made in the bone marrow, but then they are maturing in the thymus gland, which sits right above your heart. And they will make different cell types. So memory cells, killer cells, and helper cells. Memory cells, again, that goes into your long-term immune buildup. We're going to go into those into vaccinations. The T killer cells, cytotoxic cells, so cytotoxic, killing its own cells, T cells. Killer cells are actually meant to kill your own cells, not the healthy ones, the infected ones. That's what they do. And T helper cells, they are then actually stimulating B lymphocytes again. So it's kind of everything talks to everything, that immune system. It's kind of a bit complicated, but the T response is generally more attacking body cells that have become infected. If we had time, and we have time, you can produce cartoons and see if we can make cartoons out of those different immune cells to kind of show their functions.